This is the Component 2 class question book, and we're on to question 8. Germinating pollen grains. In vitro, it means literally in glass. It means in the lab rather than in the natural flower environment. And you can hang a droplet from the underside of a cover slip using petroleum jelly. So you've got you can find this on YouTube. You've got this slide there, and you hang the drop, and then you can look at the drop through the microscope. Pretty good experiment. And here's hopefully some things that you are familiar with. You might not have gone down to this magnification when you did your calculations. The smallest division on the stage micrometer measures 100 micrometer. So here we've got the stage, and so this is 100 micrometers, which is 0.1 millimeters, which is what you know already. So either from the center to the center, or from one edge to another edge. So from there to there, for example. Calibrate the eyepiece graticule and express your answer to 1 decimal place. So we can just say what we see here. So we've got how many eyepiece units? We've got 40 eyepiece units. So that is 40 EPU from there to there. So 40 eyepiece units equals 100 micrometers. That's just what we can see. And of course we don't want 40 eyepiece units, we want 1 eyepiece unit. So to get 40 to 1, we're divided by 40, so we're going to have to do the same here. So 100 divided by 40. 100 divided by 40 is 2.5. So that's our answer there, 2.5 micrometers. We are given, I'm just going to, there we are, rip this out. We are given some nice pictures. Use the eyepiece graticule, so that's our scale there, to measure the length of pollen tube indicated by an arrow from each photo micrograph. Measure each pollen tube to the nearest whole number. Enter the values in the table below. So we are just, so 0 we can't do, we are looking at here. Here's our pollen tube here. So that on our scale is 1. On this one here, I reckon the end of it's there and the start of it is there. So 1, 2, 3, 4, 5, maybe 5 or 6. I'm going to put 6. The mark scheme actually says 5, but then uses 6 in one of the calculations later on. And we've got this one here where we've got 1, 2, 3, 4, 5, 6, and then kind of 2 halves. So we'll have 7 there. Measure each pollen tube, so we've done that, and calculate the actual length. So this is in eyepiece units, so remember now each eyepiece unit is 2.5, so we're going to have to times these all by 2.5. So 1 times 2.5 is 2.5. We have got 6 times by 2.5 is 15. Measure each pollen tube to, yes, sorry. Write your answers into, yeah, okay. 15. And 7 times by 2.5 is 17.5. We could put these to the nearest whole number. So we could put 3 and we could put 18 there if we wanted, but these are fine. These are fine as they are. You could have, if you'd measured that as 5, then obviously that would be 5 times 2.5. That'd be 12.5 there. So we're going to plot a graph, and we need a suitable scale for the y-axis. Well, what do we need to go up to? We need to go up to 17.5. So choosing a scale here, trial and error. What happens if we used 1 square per micrometer? We'd have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17 and a half. That's less than halfway there. So we're going to we could double that scale. So we could go up in 2 squares. So we're looking at 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So that's the scale we're using. And plot a graph. So 0 is 0, so we've got, we better put that 0 on there. We've got a figure for the origin, we've got a point there, and then we've got one is 2.5, so time, so look, 20 minutes. 20 minutes we got 2.5, so 20 minutes 2.5, so 1, 2.5, and they give you kind of half a box tolerance on this. 40 minutes is 15, so 15 and 40. Is there pretty hard graph paper to use this, it hasn't got any kind of bold lines on it. 60 is 17.5, that's easier up there, 17, so that's 15, 16, 17.5. And in biology most of the time we're happy that we can join dot to dot with a ruler, try to get it from the centre of the point, there we go, and you draw this with a pencil. I've drawn it in pen just to make it stand out, hopefully, but if you make a mistake and you draw it in pen then you're in trouble. How could you make this data more reliable? Well, you would always the same answer. You would do more repeats and calculate a mean. So more repeats, or more tubes or more measurements, and calculate a mean. Part B. Now this is a part that students did not do well on. 4 marks, and they threw away at least a couple of marks here. Using the technique, design an experiment to investigate the effect of pH. So our independent variable is going to be pH. But it's going to be the same method. So here's the method. So we're not going to change any of that, but we're just going to change pH. So in our answer, we need to sort of talk about our independent variable, how we're going to change that. Then we need to go, okay, what about our dependent variable? How we're going to how we're going to measure what we're going to measure. And then we need usually a couple of control variables to think about, and that's what they're wanting. So we are going to place pollen grains in a range of pH, best to say buffers here, buffer solutions, as the pH might change, and you could describe, I'm going to add acid or alkali until we get the right pH, but usually using buffer solutions is the best thing. And what we're going to measure, we're going to measure pollen tube length. So we're going to measure pollen tube length, and then let's do this every 20 minutes. That can be the same, every 20 minutes. And then our control variables, well, what we're going to keep the same. So pollen from the same plant, so use pollen from the same plant, or even from the same flower. Other control variables, well, you can always say temperature here, so use the same temperature. We might even suggest a temperature, and and our other suggestions here as well. We could put use the same concentration of solution, same volume or number of pollen grains, it's the same number or mass of pollen grains, same light intensity, and so on. But the marks here for just a couple of controlled variables. Next we have some more pictures, and these are pollen tubes, and we're saying there's a bit about your cell structure here. A01 core concepts, vesicle, and further back down here we've got mitochondria, rough endoplasmic reticulum, Golgi body. I wonder what they're going to ask us about if they're labelling those. So we're relating what we know about pollen tubes and how they grow to the stuff in them from the core concepts. Describe the role of the vesicle and the labelled organelles. So it's like a mini essay, isn't it? So we're going to say our structure is vesicle, and we're going to go mitochondrion. Not that I can spell it right. We're going to talk about rough endoplasmic reticulum. Then we're going to talk about Golgi body. So you can see where these four answers, we're going to have our four separate points for these. So we're going to say, releases digestive enzymes. Releases or contains digestive enzymes or hydrolytic enzymes. Protease, exocytosis, e.g. protease by exocytosis. That's kind of a full answer. It's going to hydrolyze a path through the tissues, without space there, but that's another point there. Mitochondria, well, they only ever do one thing. they're going to release ATP energy through aerobic respiration. Release ATP energy by aerobic respiration. Rough endoplasmic reticulum, or protein synthesis. So we can have any example of proteins like enzymes or transport proteins. So we can have protein synthesis, e.g. those hydrolytic enzymes that we were just talking about. Golgi. Well, our go-to answer for Golgi is modifying proteins. We could say packaging proteins, or making these vesicles, package them into vesicles. So modifies proteins and forms a vesicle. So hopefully when you see a question like that and you go, yeah, it's just asking me for a load of knowledge, it's actually down as AO2 because you're using the diagrams and then relating your knowledge to it, but this is just stuff from your notes. So 80 marks, maths and remembering some stuff.